Assalamualaikum warahmatullahi ta'ala wabarakatuh, and a very good day to everyone. My name is Muhammad Tarmizi bin Muhammad Noor, and I was supervised by Dr. Wan Ibrahim binti Wan Osman. The title of my study is Cytotoxicity Effect and Morphological Changes of Methanol Extract of Chrysanthemum indicum on HL60 cancer cell line. So let's dive into the contents of this presentation. First, I will brief on the scope of my project, and then the methodology of this project, the expected result, and also Gantt chart and the progress for this final year project. So the objective of this study is there are two, general and specific. For the general, we are going to determine the cytotoxicity of Chrysanthemum indicum on APL cell, HL60 cell line, and there are four specific objectives that has been constructed for this study. The first one is we are going to extract flowers and buds from the Chrysanthemum indicum by using methanol extraction. Then we are going to evaluate the cytotoxicity on buds and flower of Chrysanthemum indicum at different incubation period, 24, 48, and also 72 hours. And then we are going to determine the apoptotic inductions of bud and flower of Chrysanthemum indicum towards HL60 cell line by using annexing 5-FYP staining. And lastly, we are going to determine the effect of flowers and bud extract of Chrysanthemum indicum on the morphological changes to the HL60 cell line. There are two types of hypotheses. The first one is alternative hypothesis. There is a cytotoxicity of flower and bud extract of Chrysanthemum indicum on HL60 cells. And for the null hypothesis, there is no cytotoxicity of flower and bud extract of Chrysanthemum on the HL60 cell lines. So for the problem statement, from the literature view, it is found that there is a risk associated with the current treatment, arsenic trioxide, and the combination with the all-trans retinoic acid with the treatments on the APL patients. It is because from the literature view, it found that liver function as the major sites for demethylations of arsenic. That is why ATO will increase the mitochondrial reactive oxygen species and also increase the risk of liver injury. There are also adverse effects of chemotherapy treatment, such as nausea, vomiting, and so on. So, natural bioactive compounds that may encourage anti-leukemic actions or anti-cancer are very crucial for us to improve the effectiveness of the treatment or reducing the adverse effect from the treatment. So the study design, firstly we are going to try the Chrysanthemum indicum samples, and then extraction is being done with the methanol. Then for the cells, we are going to use PMI medium, Roswell Park Memorial Institute 1640 medium, and then we are going to treat the samples on the cells. And for the MTT assay, to know the inhibition concentrations after the incubation at 24, 48, and also 72 hours. Apoptotic assay is done by using annexing 5-FYP or PI kits, and morphological study by fluorescent microscope, and lastly will be the data analysis and report. So this is a bit of the literature review from the my reading. Anti-cancer properties of Chrysanthemum indicum, such as the Chrysanthemum may induce apoptosis by separations of constitutive step 3 activation in human prostate cancer, the U145 cell line. Besides that, it also inhibits neoplastic transformation by blocking activations of the MAPK pathway and stimulation in cancer. Okay, next. Acute myeloid leukemia. Acute myeloid leukemia is a bone marrow malignancy where hematopoietic precursors are interrupted at such an early developmental level. And from the French American British FYP, classify APL as in M3 subtypes, and cytological characterizations of APL is by dominance of promyelocyte cell. That's why it's called acute promyelocytic leukemia. And APL is caused by the translocation between chromosome 17 and chromosome 15. Until now, there are available treatments include the chemotherapy and also what I have mentioned before, TO and also ATRA. So the methodology for the extraction, the first step we are going to do is the drying and also grinding the sample to produce a fine sample so that it is easier for us to soak the sample into the solvent, and then solvent will be added and left for 24 hours, and then the extract will be centrifuged to remove the impurities before the solvent is removed by rotary vapor at 50 degrees Celsius. So the final extract will be collected and will be filtered several times and freeze dry, then keep in the refrigerator for future use. So this is the formula for to calculate the percentage of yielded extraction from the sample. For the cell culture, the first one is cell thawing. Cells is going to taken out from nitrogen tank. Then we are going to culture the cells on the complete medium. After that, monitoring the cells and also subculture the cells before we seed the cells into the 96 well microplate. So these are one of the formula for the calculations of the number of collected cells in the well. For the cell inhibition, samples are treated to the cells according to different concentration, 12.5, 25, 50, 100, 200, and 400 microgram per ml. And MTT is used to measure the living cells by forming the blue formation. And as you can see from the picture there, the darker color resembles the higher concentrations of cells that are lived in the well. So IC50 can be determined by plotting the graph of HS, its cells inhibitions against the concentrations of the sample. So the apoptosis is done by following this step, and flow cytometry analysis and fluorescence microscope analysis will be run within 1 hour of the test. So apoptosis, how to interpret the flow cytometry graph. Actually, from the graph there are 4 quadrants: upper left, upper right, lower left, and lower right. And each quadrant resemble different conditions of the cell. For example, upper right resembles the late apoptotic or necrotic cells, and the lower right resembles the early apoptotic cells from the test, and also the lower left of the graph resembles the cells that are alive from the test. Okay, expected result. This is just the example, diameter table for the incubation period from the inhibition considerations of HSCT cell lines from MTTSA. So for the Gantt chart, to now, January, there is still one component of planning, which is the lab working. We are not allowed yet to enter the lab. We are hoping so we can enter the lab as soon as possible after this final year examination. For the revision from previous presentation, problem statement is adjusted by stating why chrysanthemum is selected in the study and the relationship between side effect and chemotherapy, the arsenic in uses in treatment for liver injury. And the second objective is replaced with the words evaluate. The uses of phytochemical properties is replaced with phytochemical constituents, and tables are constructed in the expected result to resemble the extraction yielded and also inhibition concentrations of extract towards the cells. So there are also questions from the panel. For example, what are the primary metabolites and differences with secondary metabolites? So primary metabolites is basically metabolites that are required or essential for the reproduction or the growth of the plants. And secondary metabolites, other metabolites that are not required for the growth for the plants. And what is meant by SC50? SC50 is the half maximal inhibitory concentration. That is, we can measure of the effectiveness of a substance in inhibiting a specific cells. In this study, it will be HSCT cells. And why methanol is used in the extraction? Actually, from the previous literature review, there are also several studies that using several solvents for evaluating yielded extract from the extraction, and it is proven that methanol is the best solvent among others. I think that's all from me. Thank you for your attention. Assalamualaikum.